Our today's topic is Prion Hypothesis. Prion Hypothesis claims that a protein behaves like a living microorganism to infect an individual by various routes, survive metabolic clearance, self-replicate in the body, reach the target organ, induce a cascade of neurodegenerative damage resulting in disease and ultimately death of the infected individual. 43 years have passed since it was first proposed that a protein could be the sole component of the infectious agent responsible for rheumatic prion diseases. So, over decades of research, many milestone discoveries have provided crucial evidence in favor of this prion hypothesis. Some key experiment in this field, like Alper and coworkers in 1966, demonstrated that procedures that obliterate nucleic acid, such as very high doses of ionizing radiation and UV radiation, didn't destroy the infectious material. This study also reported that the minimum molecular weight to maintain infectivity was too small to possibly be a virus or any other type of microorganism, right? So based on these observations, speculations arose that infectious agent might be protein based, right? In 1982, Prusiner and co-workers coined the term prion for these particles. Then the protease-resistant prion protein, PRPSC, was isolated from the infectious material by these scientists and also showed that PRPSC concentration was proportional to the infectivity titer. Right? So these breakthroughs radically changed the prion field and much effort then turned to study this protein. Infectivity was shown to be retained in highly purified preparation of PRPSC, in which no other protein was detectable. Although sample did contain lipids, carbohydrates, and perhaps nucleic acids also, it was indicated that prion proteins can exist as both a normal cellular protein, that is PRPC, PC, and pathological isoform, PRPSC. This we have already discussed in our previous lecture. So no chemical differences can be detected between the two forms of proteins. So the cell-free conversion of PRPC into PRPRAS catalyzed by the pathological protein also provided the support for this prion hypothesis. Now, what is this PRPRAS? This is protease resistant prion protein, right? So in this system, which was developed by Kogi and co-workers, what they did, they purified PRPSC mixed with purified PRPSC form and produced a low yield of PRPRAS formation under non-physiological conditions that precluded infectivity studies. In 2001, a new system for in vitro replication of prion was developed then. It is termed as protein misfolding cyclic amplification, PMCA. The in vitro generated material then maintains all the biological, biochemical, and structural characteristics of in vivo produced prions, right? So these findings confirmed the central tenet of prion hypothesis, which is that prions can propagate indefinitely and that a newly generated PRPSC triggers further misfolding to produce an autocatalytic process of prion replication. Now, I will tell you about some major milestones in the prion hypothesis. In 1937, accidental transmission of disease scrapie in the sheep. Now, what is scrapie disease? I have already told you in our previous lecture. So then in 1939, scrapie disease was transmitted experimentally by scientists, right? Then in 1966, it was demonstrated that these small size of infectious agent responsible for this disease. Then Kuru disease transmitted to chimpanzees in 1966, right? So what is this Kuru? Actually, word Kuru means shivering or shaking, right? So Kuru is a neurodegenerative disease caused by these prion particles, infectious particles, right? So Kuru actually, it's an island, means a nation of Papua New Guinea, is one of the most fascinating countries in the world. So Kuru was found to affect a tribe called Four, right? So four people means people living in this region ritualistically cooked and consumed body parts, mainly brain, of their family members following their death. Why? To incorporate the body of the dead person into the bodies of the living relatives. This means helping to free the spirit of the dead. So brain is organ enriched with these infectious prion. So people consume body part, mainly women and children, had much likelihood of being infected than men. So Gazetzik and Gibbs in 1966 were successfully transmitted this disease to chimpanzees, right? So this is how Kuru transmitted. So in 1967, infectious agent not affected by DNA elimination, it was confirmed. Then protein-only hypothesis was proposed by Griffith in 1967, same year, and in the same year, prion protein isolated from the infectious agent. Then in 1982, Prusiner coined the term prion for these particles. Then identification of PRP gene, right, in 1985. PRP transgenic mice 12 neurodegeneration um, in 1990. PRP knockout the mice resistant to infection in 1993. Then discovery of yeast prions in 1994. Reed Wigner proposed that yeast proteins URE2 and SUP35 behave like prion. So it was discovered that prion-like particles also present in yeast in 1994. In vitro generation of yeast prion then 2000 efficient prion replication in vitro by PMC in 2001, then production of synthetic prions in 2004, and generation of bona fide infectivity in vitro by propagation of PRPSC in 2005. In vitro generation of infectivity with defined components in 2007, then de novo production of infectious prions in 2007, and in 2010 in vitro generation of infectious material from recombinant PRP, right? So these are some major milestones in this prion hypothesis, right? So if any question, just write in the comment section so that I can answer. Thank you so much.